Sanger sees itself as an ideas factory. First of all, through our scientific programs, of which we have 4, covering a wide variety of different life sciences projects, from single-cell sequencing for the Human Cell Atlas, to cancer genomes, to the fairly ambitious aim of sequencing the genome of every single species on the planet as part of the Earth Biogenome Project. And we see ourselves as staying in that leadership position. And the way we do that is through these other approaches. So we want to be able to see ourselves taking calculated risks in adopting new technologies flexibly and capabilities as they come along. If we just sit on the coattails of others, we're not going to get very far. And it's all about large scale. Sanger's expertise has been about scale since the beginning. We've always been pushing the edge of what we can do there, both in terms of lab processes and in terms of IT and informatics in processing that data. So our platforms that actually deliver this stuff are divided into 2 sort of large strategic areas. The first one is what we call scientific operations, which delivers large-scale, high-throughput platforms for the rest of the institute, and also our IT services, which are another core platform. We don't just do sequencing, as the title of this festival is, and biodata as well. So we are expanding beyond just sequencing into other forms of single-cell analysis, imaging, and so on and so forth. But for today, I'm focusing on the genomic sequencing. In terms of our IT provision, and you'll forgive me for going in there a little bit because that's my world, we have traditional HPC services. For several years, we've been moving towards a cloud-oriented way of delivering our science, which means we have a sizable on-premise cloud as well as our traditional HPC environment. We have got extensive experience now, of course, in handling data at multiple petabyte quantities. And our IT services have a large amount of expertise in informatics, which we can use to help the scientists and the informaticians within the scientific programs with their scaling of their applications in particular. It's all very well developing your algorithm on your laptop, but when you now want to run this on 500,000 samples, it's a different world. So I'm not going to speak to this slide much at all, because I'm sure Sir Rory is going to tell you far more eruditely all about UKBiobank than I can. But I think it's just worth pointing out that this is a really good example of what we've heard about earlier today. It's collaboratively funded by the government, industry, and ourselves, the Wellcome Trust rather, and provides this link between the genome sequence and the phenotypic data. So where did the main phase project come from, and how did we get involved in it? So it started with a Vanguard, what they call the Vanguard Project, which is we were asked to sequence an initial 50,000 whole genome sequences, just to establish that it was a feasible project. And that's more or less complete now, and we've done that successfully. And then this consortium came in to fund doing the remaining 450,000 samples. Now, Sanger was delighted to be invited together with Decode Genetics in Iceland to perform this as a joint enterprise between the 2 of us. There are some interesting changes between this and the Vanguard project. So in the Vanguard project, the analytics was a separate contract, which was awarded to Seven Bridges. For the main phase, the project includes the informatics as well. And it also includes a nicely challenging, and I said we like challenges and pushing the boundaries, doing a joint variant call on all 450,000 samples, which I'm sure you're aware is not a trivial undertaking. And the timescale for this project is also quite challenging. We were asked to deliver the first 120,000 samples across both partners before, and the joint variant call before May 2020. So if you work that out, that means we kind of have to have sequenced it all by sometime in March to give time to do the variant call. And given that the project started in September, that's quite an aggressive time frame. So, how have we scaled that up? Because that's actually a much bigger scale even than Sanam was used to doing in the past. So, well, Illumina helped us out because they managed to drop 7 new NovaSeq sequences inside 2 weeks, which is nice. And we got on with our IT providers to start building some dedicated infrastructure. Because prior to this, the Vanguard project had run on our internal private cloud, shared with our regular research science, but in a secure project within that. But this is a bigger order of scale, so we now need to be right. Now we need to do this dedicated. This is a production process that's going to run for a couple of years. Keep it separate from the research environment completely. When you've only got 6 months to deliver that first piece of work, and you've got regular lead times for procurement and so on of 3 to 4 months, how are you going to deliver roughly doubling your compute capacity and your storage capacity in that period of time and still get all the sequencing done? Technically, we could do it, but the time was a problem. So, that was why the decision was arrived at to continue partnering with 7 Bridges, and we'll talk about that in a minute. But we are now at the scale that we need, and we can, sequence 1,000 human whole genomes with every load of the sequences, and we do that 3 times a week. So that's 3,000 samples a week. And one of the things I'll come to is the infrastructure is designed so that we keep pace with the sequences. We're not just accumulating data and then, well, we'll get around to analyzing it. No, we are analyzing it at the same speed we're generating it. So just to put that scale into some sort of perspective, we all love exponential growth graphs, right? So you can see here that the scale is getting crazy. Even when we first started next generation sequencing, about there, it's not even a pixel on this graph. So, and you can see just since where Vanguard started, we've more than doubled since Vanguard started, and that was only a year and a half ago. And we sequenced more DNA in the past year than we've done in the previous 25 years. So it is possible to push the scale like this. So what does the pipeline actually look like? Well, first of all, we were fortunate that my informatics team and IT team, particularly led by Peter Clapham, had built this on-site OpenStack private cloud, which allowed us to get started very quickly. And that has been used, as I said, to run the Vanguard process. That's actually a picture of it on the right. That is the actual cloud cluster itself. I can't resist putting a picture of the IT kit in. But simultaneously, of course, we've got to scale the lab approach, which is actually a relatively small number of sequences. We can deliver this on our half of it with only 15 NovaSeqs. And Sanger already has experience from the very early days of, at times, operating more than 100 sequences. So the number of sequences isn't so much the challenge, although you can imagine that there is, with this number of samples coming through, there are some interesting logistical challenges with sample management and just getting the reagents through stores and procurement and making sure everything comes in is interesting and fun. But to do the analysis, we have come up with this hybrid cloud approach as a result. So we reduced the scope of our on-premise cloud that we were building to just enough to make sure that we've done some initial QC, we've merged all of the samples from the different runs, so that we've got a sensible, unaligned cram file format that we can hand over to 7 Bridges for further analysis. The advantages of this approach are, firstly, that it offers some continuation as well with what we did for Vanguard. 7 Bridges already had the pipeline. And secondly, it hid all of that procurement latency, so we could get started straight away. And this is roughly what the pipeline looks like. It's probably not terribly readable, But everything that's blue is us. Everything that's red is 7 Bridges. So you can see that we perform the sequencing and the analysis in the initial QC, top left, with a target time of 48 hours. The sequences are reloaded every 48 hours, so we have to get that data done and out the door within that 48 hours. It's then passed down to 7 Bridges, where this is all running on the Google Cloud at the moment. Doesn't have to, but that's where it is today. And again, they have a target of delivering the variant call within 48 hours, and that's then sent out to the final repository that UKBiobank have appointed, which is the EMBL EBI. So actually it goes to the cloud and then back to the Wellcome Genome Campus again. And then finally there will be the joint variant call at the end. So this is a huge project, as you can imagine, for us. It uses 5 times as much sequencing capacity as the rest of the Institute's science put together, which gives you some idea just how big this is. And it has touched almost every part of the Institute. Every part of the Institute that isn't the direct science, actually. There is some scientific involvement from human genetics and so on, but mostly this is actually a project that's being delivered by the underlying platforms groups. There's some contact details for you to take there if you want to talk to, well, me and my team about the IT side of things. Ian Johnston, who unfortunately couldn't be here today, he was supposed to be, but if you want to know all the nitty-gritty about how you actually do the physical sequencing at that sort of scale, Ian's your man, and Cordelia can tell you all about the overall project, since she is the overall boss of it. So at that point, I will hand over to Sanam, who can tell you all about 7 Bridges side of it, how they're contributing, and how that helps us. So thank you very much.
Hi everyone, I'm Sanam, and I'm a senior bioinformatics scientist at 7 Bridges. So today I would like to talk a little bit about what happens behind the scenes from where Tim left off, and how we also managed to scale to address the needs of the UKBiobank's world-leading whole genome sequencing program. But before I jump in, I would like to start with a little bit of history of our involvement in this program. As Tim briefly mentioned, we have been selected almost 2 years ago as the informatics provider for the Vanguard phase of the program, which intended to inform the main phase and reveal the specific challenges that can be only apparent when you start processing at this scale. So we initially started with processing 5,200 samples, then we proceeded with the remainder of 44,800 samples, bringing a total number of about 50,000 samples as part of the as part of the Vanguard phase. But while we are working towards this aim, Sanger has approached us to partner in the main phase of the program, where with Decode Genetics and Sanger, we are collectively aiming to sequence the full cohort of the UKBiobank, so bringing it a new set of 450,000 whole genome sequences to be completed within 2 years. So as part of the main phase, currently this is how the flow looks like. UKBiobank sends the samples to Sanger, who completes the sequencing and the initial quality control analyses. Then Sanger sends us unaligned CRAM files and the results of those initial quality control reports. We do then ingest these data to the 7 Bridges platform, where we process them at scale using BWA, GATK. We do perform quality control analyses across various stages of the analysis pipeline, as well as we perform data management and storage for Sanger. Subsequently, we deliver daily and monthly reports back to Sanger to inform their sequencing team on what to sequence next. And for the samples passing through all the quality control thresholds, we do deliver aligned CRAM files, GVCF files, as well as quality control reports to the designated UKBiobank's designated data archive. So we do run all of this operation on the 7 Bridges platform, currently deployed on the Amazon Web Services Frankfurt region. However, as the needs of the program grew, we rapidly increased our processing capacity using our multi-cloud capability. and Google Cloud Platform's both London and Belgium regions. Currently, the informatics for the main phase consists of multiple workflows that are built following industry standards. Unaligned cram files received from Sanger are aligned against GRCH38 with a workflow developed following functional equivalence standards, as well as Broad Institute's best practices for data preprocessing, as well as germline short variant discovery. So one of the core values at 7 Bridges has been always reproducibility. So when we are building and implementing these workflows, we have followed 2 open standards in this case, which was one of the most common workflow language, where you can describe tools and workflows in detail with their inputs, all the parameters and outputs. And the second one was Docker, where you can store tools, associated libraries, as well as their runtime environments. So by using these 2 standards, we have ensured that the workflow built for the program is fully reproducible and always completely portable. So once the data gets released, the researchers and approved researchers of the UKBiobank can actually pick up the exact same workflow and run against using their own input data and harmonize their own data to this massively large data set. And this workflows, how it looks like, is like this, and it's a rather complex workflows. And they're built using an intuitive and visual workflow editor on, again, 7 Bridges platform. So this interactive interface allows our bioinformaticians and users to simplify their common workflow language development experience significantly, as well as reduce the time for development and testing. And this platform also contains an inbuilt version control system, which ensures that whenever you make a change on a tool or a workflow, a new version is created, and the execution of all of these samples are tied to specific versions of the workflows and the tools. And Tim mentioned and showed in his diagram, there are multiple breakpoints and multiple checkpoints on quality control metrics. So we need to ensure that samples go through these checkpoints, and if a sample fails a prior checkpoint, shouldn't proceed along the next step of the pipeline. So therefore, there's a certain need of complex orchestration, which we achieved by using 2 core technologies, which is our native API, as well as the automation development kit, which allows users to rapidly create their own automated solutions. Using these technologies, we are currently managing to deliver all samples we receive from Sanger, process them, and deliver to UKBiobank, which comes about roughly over 11,000 genomes per month. And we achieve over 11,000 genomes using automated workflows, which reduces human error as well as increases throughput, because you do not need to have any manual intervention. But as I also mentioned previously, one of the key factors was the multi-cloud. So where we can rapidly utilize computational resources across different cloud providers as well as different cloud regions through a single pane of glass view. So you're looking at the same interface, you're using the same API, nothing changes, but you're executing across multiple different providers such as Google and AWS, and across different regions such as Belgium, Frankfurt, or London. And while you're doing that as a developer, you only develop the workflow once. And the platform automatically manages that movement of the data, movement of the tool or a workflow for you to bring the scale, speed, and the cost that is needed for a program of this scale. And while you're doing this multiple infrastructure and multiple region analysis, reproducibility becomes more and more important. And in order to ensure that, we do provide services such as detailed logging. You can take a look at task logs, how a specific tool is executed, how the workflow is executed, and you can have a tight connection with the specific versions of tools and workflows, as well as we do store provenance information. So if there is a result file created and delivered to UKBiobank, we can always go back to the specific execution of the workflow and identify the actual logs of the workflow, as well as operational information such as when this execution happened and who initiated that execution. And while we're doing that, again, Tim showed multiple very compressed timescales that we are running against, ranging from 12 hours to 48 hours across different stages of the full analysis workflow. And you have to achieve this timescales while managing the cost and controlling the cost. So we do again achieved by using technologies such as our native spot and preemptible instance support and automatic retries where those instances may fail. And we do employ additional technologies such as scattering and scatter-gather technologies to divide the analysis into pieces, then gather together. And lastly, but probably the most importantly, the expert scientists, so the scientists at the core of this whole operation. So our scientists that are expert in large-scale genomics analyses have partnered in this project with Sanger scientists to deliver this world-class resource that is UKBiobank whole genome sequencing dataset. So this dataset is currently going through a variety of stages of analysis right now, but UKBiobank has already announced that the first tranche of the data is expected to be complete by May this year, and after 9 months of exclusive access period, it will be released to the all approved UKBiobank researchers. And the full data access and distribution is managed by UKBiobank. So if you're interested in this unparalleled resource, I would strongly suggest you to take a look at their website at ukbiobank.ac.uk. With that said, today Tim and I have talked about UKBiobank's whole genome sequencing program, how Sanger utilizes its unique experience and decades-long experience in human genetics and sequencing, how Seven Bridges uses its extensive capabilities and experience in large-scale bioinformatics to realize the aims of the UKBiobank's whole genome sequencing program. But for those who may not be familiar with us, I would like to finish by mentioning a few of our other capabilities that we employ at Seven Bridges, such as our analytics platform that provides access to over 400 bioinformatics tools and workflows that are optimized to run on cloud, or our unique algorithms such as Graph Genome that eliminates the reference bias in sequencing experiments, or we do provide out-of-the-box access to the world's largest genomic data sets such as the Cancer Genome Atlas, Target, CPTAC, or ICGC. With that said, if you'd like to learn more about us, our work with Wellcome Sanger Institute, please drop by our booth number 6 or join us at the live lounge talk where my colleague Amit Jain will talk about our graph technology for the future of genomics analyses. Thank you.